Monica Gupta, your TA for the session, and today we'll be discussing about week four's lectures. And the session will go the way it has been going in the last 3 weeks. We'll first have a recap of what you guys have studied in the last week, and then we'll take up the questions, the assignment question, and some of the questions that I have brought for you guys. Okay, so let's start. Okay, so are the slides visible to you all? I hope they are. So let's start. So in the last week's lecture, we did something in the continuation of what we were doing in the week three. So basically, the genetics basically is what we were studying. So here we have taken up a common example, that is C. elegans. So we'll see it according to examples throughout the lecture. First of all, understand what is mutant isolations and why it is important. So mutant isolation is basically as a procedure is done to understand the function of a particular gene. If a gene gets mutated, then obviously the function it was supposed to perform will not be completed, and it won't be able to perform that function with that perfection that it was supposed to. So obviously that will affect that particular character, and that is how it will help that what gene is important for which character or which process happening inside the body, to better understand the developmental biology of that particular organism. So, what happens in mutant isolation is the cell fertilization allows quick recovery of recessive mutations. ओके, सो रिकवरी जो है रिसेसिव म्यूटेशंस की, अगर सेल्फ फर्टिलाइजेशन होती है तो इट इज रिकवर्ड बेटर. And the number of chromosomes set screened is twice the number of F1 progeny. Obviously, it will be twice the number of F1 progeny, because after the self fertilization F1 progeny, the number of worms or whatever organism will get, the progeny we will get is obviously will be the double. So, the set screen will be twice the number of the F1 progeny. The mutant here used is ethyl methane sulfonate, EMS in short. So EMS, how to use EMS and what amount of EMS is sufficient for causing the mutation, what amount of worms we have taken. So all of this is what hit and trial method we have to check it, that what percentage of EMS is working for us, and obviously from the literature also. So EMS is the amount of EMS that is supposed to be given to a particular number of worms, or to the organism on which we are studying, is according to the literature or some hidden trial. We have to devise that. It is not some written rule somewhere for how to use EMS and what amount of EMS must be used. Okay, so 1 in 2000 gametes exposed to 50 millimolar EMS will carry mutation in an average gene, and screening 10,000 gametes is usually sufficient to recover most common loss of function mutation. So they are suggesting that nearly 10,000 gametes is what is sufficient to recover most of the common loss of function. Loss of function means whatever gene which is functional in its wild state, and when the mutation happens on that gene, in that particular gene, then that gene loses its function, it won't be able to perform its function. So that is what loss of function means. The opposite term to loss of function is the gain of function. So what happens is a gene in wild type situation is generally um, inhibited, but when a mutation happens on that particular gene, then in that case it will uh, gain the function. It will be functional, that functional in a way that it was not required in wild state to function that way, but when gain of function mutation happens, it will start performing that function when it was supposed to switched off. it will stay switch on. So that is a gain of function mutation, which is again a problematic situation because some genes needs to be shut off, they needs to be switched on whenever required, not always. So gain of function will do that, it will keep the gene on always, that will again cause a problematic situation. uh, so this was just a small experiment they did to understand the genes involved in vulva formation. So when they were selfed, the plus over plus situation was in the parent PO generation was selfed, was mated with each other. Then in F1 progeny we got one mutant over plus, so that means and when these mutant over plus was again self mated, then we got three types of progeny: plus over plus, M over plus, and M over M. So M over M were the mutant ones. So by this method, over 100 mutants were identified. So this is a very important method to identify the mutants. Two general classes of phenotypes that can occur because of mutations. As I told you, it can be gain of function, it can be loss of function. If it is a loss of function situation, it will be vulva-less, and if it is a gain of function situation, it will be multivulva. So there can be two types of phenotypes that can be seen due to the mutation, either multivulva or vulva-less situation, depending upon what kind of a mutation is happening in that particular gene. And what is complementation? So what happens in is, as we have seen in this heterozygous situation, we got M over we when we self mated this M over plus, we got plus over plus and M over plus. So this M, the mutant part, got complemented with this wild type so that it could show its general wild type expression. So this is complementation, when a mutant allele gets complemented with the wild type allele and that wild type allele takes over the function and the mutant expression doesn't comes out, it is the wild type function that comes out and that is what expresses. Okay, so that is complementation. So the 100 mutants represented nearly 20 genes, 20 different genes, which could lead to these type of expressions, multivulva or vulvaless. Okay, so let's move towards what next is. So the detailed observation of mutants using Nomorasky optics. It is a method to understand these mutations. So genotype is wild type, phenotype is again wild type, and the fate of PNP cells. PNP cells are those cells which are involved in the formation of the vulva. The whole six, I guess six, yeah, eight cells are those, and those eight cells are involved in the formation of the normal vulva in the C elegans. C elegans is the example we are taking here. Okay, so genotype is wild type, phenotype is wild type. Every cell will perform its function, whatever it was supposed to perform. An invariant pattern will be seen because the cell nearest to anchor cell will be the primary, will perform the primary fate, then the lateral cells will perform the secondary, and the farthest cell will undergo the tertiary kind of fate. Okay, now genotype is lin 26, phenotype is vulva less, so no PNP cells will form, PNP cells होंगी ही नहीं यहाँ पे. ठीक है, when the genotype is lin 3, phenotype is again vulvaless, and all of the cells will become tertiary in expression, induced fates. So that means lin 3 is the one which induces the fates of the cells. It is the inducer which induces the tertiary fate in the cells. So uh, if I'll simplify it for you to understand that, that means that this secondary cell, this P5P cell released this lin 3 to induce these cells to perform the tertiary fate, to undergo the tertiary fate. Okay, Lin 15, it causes the multi-vulva type expression. So it causes what? It causes the uninduced fate. Basically, it will stop the, it will be the inhibitor of the mutation induction. So it will not lead to induced fate, it will lead to uninduced fate. That means these cells will form an alternate 2 1 2 1 2 1 kind of a pattern, because the induction to form the tertiary kind of fate didn't happen because of this Lin 15 type of genotype, and it will lead to the formation of multi-vulva. Okay. Let 60, it is a dominant kind of a genotype, so it will again lead to the formation of multivalva, and it will again, we will not be able to conclude anything from this kind of a mutation. ठीक है, तो डोमिनेंट म्यूटेशंस क्या होती हैं? इन्फरिंग फंक्शंस ऑफ डोमिनेंट म्यूटेशंस आर नॉट इजी. This will require further experiment, obviously, but dominant mutations are those mutations which will dominate the wild type expression. Whenever this kind of a mutation is present, then the wild type of allele will not be able to complement that expression. The mutation will be dominant over the wild type of an expression whenever this kind of a mutation is seen in the organism. Okay. So it can be of any type, it can be hypermorphic, it can be neomorphic, it can be entomorphic. So what does this hypermorphic means? That it will lead to an excessive expression of that particular type, for example the multivalva kind of an expression. So these dominant alleles can either lead to this hypermorphic and excessive expression, a hyper expression of that particular type, or it can be neomorphic type of mutation. That means a new kind of expression can be seen, a new kind of a trait can be seen, so that is neomorphic. Next is the antimorphic. Antimorphic means it will show some opposite kind of a fate that it was supposed to show. So in hypermorphic, a hyper expression will be seen. In neomorphic case, a new kind of an expression will be seen. In antimorphic, and opposite kind of expression will be seen in case of the dominant mutation. So these are the three types of mutations that can cause these three type of situations in the organism. ठीक है, so usually isolation of recessive loss of function alleles will also be required, because that will help us to understand these mutations better. Cloning genes by mutation. So there are three steps included in this cloning genes by mutation. Correlation of the genetic and physical maps near the gene of interest, DNA mediated transformation to identify rescuing clones, and identification of DNA lesions associated with mutation in the gene. So these this cloning genes by mutation can help us should follow these three steps. The correlation of genetic and physical maps, it should DNA mediated transform, it will help us to understand the DNA mediated transformation to identify rescuing the clones. Identification of DNA lesions. DNA lesions are basically some kind of a nicks, inversion, in transversion, or if there is some kind of a damage present in DNA. So these are the DNA lesions associated with the mutation in the gene. Okay. So again, genetic mapping. If you remember, in the last class we discussed the term centimorgan. So centimorgan is equals to the 1% of recombination frequency. 1 centimorgan is equals to 1% of recombination frequency. That is what they have shown here. Ki how to map some unknown genes in the genome. So how do they do that? They do that by exactly pointing the location of marker genes, and by these marker genes we will be able to determine the position of a particular unknown gene. So it can be a two factor analysis, it can be a three factor analysis. So in two factor analysis, what happens is recombination frequency between the new mutant and a known marker mutation is understood, and that is how the distance is told. And in the three factor analysis, sorry, recombination frequency between the new mutant and two different known marker mutations is understood. So obviously the three factor analysis is better than two factor analysis, because that will help us to understand the exact position of that gene with respect to these two marker genes, which we already know the location of. So this three factor analysis is the much preferred one compared to the two factor analysis. Okay. The example of cloning genes by mutation is the genetic mapping positioned let 60 between DPY20 and left the breakpoint of NDF27. This is an example of this cloning genes by mutation. Cosmids spanning the region were tested by injections for rescue, and three overlapping cosmids were found to rescue. So this is just to show you how different mutations can lead to these different expression, and how rescue of uh, in case of how cosmid helps in the rescue. Okay. Rescue with the smaller fragments of two overlapping cosmids. This encodes a protein with strong homology to RAS. RAS is a signaling molecule, a very important signaling molecule in the body. This you will understand when we'll be studying about the signaling, or if you guys know about signaling already. So RAS is one of the very important signaling molecule. Okay, next is the suppressor screen. Sorry. So powerful method to isolate additional genes that function in a particular process. So basically what they want to say is if there is any process, if there is any development going on in the body, then there is no hard and fast rule that only a single gene will be controlling that. No, there can be many genes involved in that particular function or that particular process. So suppressor screen is the method through which we identify how many alternative genes are there, are involved in that particular function or that particular expression or organ development. Okay. So this is a powerful method to isolate additional genes that function in a particular process. There can be more than one genes involved in a particular process, and suppressor screen is one such method which helps us to identify how many or which genes are involved in that particular process occurring inside the body, occurring during the development, whenever. Okay. Sorry. So the key is to design the screen such that the large number of mutagenized gametes can be observed. So this is multivalva mutation, the isolation of suppressors of multivalva mutations. They have shown the process they have taken. Multivalva parent, and another is a wild type parent. They have self them, and in the F1 progeny, we got a mutant with a wild type situation and multivalva type of situation. And then again in the F2 progeny we get different combinations, and from this we could identify that there must be another genes that are also responsible for multivalva expression. How did we understand that? Because in this is a multivalva situation, but there in if we'll see in that scenario, in this scenario in which M over M is there, so that means there is some other gene also which is causing this multivalva situation, instead of only single gene which is involved there. So this is the key to design screens such that the large number of mutagenized gametes can be observed. So suppressor screening should be done that ways only, in that way, so that a large number of gametes can be observed and we can identify the key players or the many players that are involved in a particular process. Several suppressors for lin 15 and lead 60, special alleles of new genes not recovered in the earlier screen because they have lethal or null phenotypes. So this kind of a mutation can also happen, which can lead to a null phenotypes, or we can say a lethal phenotype. New alleles of known genes, for example, LET60, several suppressors of lin15 multivalva mutations were new alleles of LET60 with a valva-less rather than original multivalva phenotype. So, the genes that were involved in the multivalva mutation, they were found to be alleles of LET60 with the valva-less expression also, rather than sticking to only multivalva type of phenotype. So this is how suppressor screen helps, that whichever genes are involved in a single process. So now there are three types of LET60 alleles: complete loss of function, the null genes, the null alleles we were talking about, the lethal alleles. Then the antimorphic, the dominant negative, which can induce the fate valvaless, or hypermorphic, which can cause a constitutive expression and leading to a multivalva formation instead of a single valva. So these three type of LET60 alleles can be present, and that should be screened through suppressor screen. Okay. So again they have shown here NOS1 and NOS2 activity in fertile. It is equal for both. In another situation, the NOS2 expression is less, and in other situation NOS1 expression is less. So obviously the NOS1 is complementing the NOS2 loss, and NOS2 is complementing the NOS1 loss. And when both of these expression is low, in that case the animal or the organism will be sterile, otherwise it will be fertile, because it has the compensation by other allele. Okay. So like suppressor mutations, synthetic mutations help us identify genes that are functional in a single pathway, the multiple genes involved in a single pathway, so that they can complement the function, they can support the process, and many other factors. Now, after this, we come to epistasis analysis. So, epistasis analysis is basically, if you guys have studied biology before, then you guys must have heard this term epistasis, which is generally used when we are studying the genetics, because in this it is not always white or red kind of expression, there can be something in between also. The one gene will be epistatic over the other. It can be a recessive epistasis, dominant epistasis. There are many kind of an expressions that can be there. So one gene can be showing its effect on another gene. It can be a trans effect, it can be a cis effect. There are many things that can be possible in case of epistasis. So it helps in the determining the relative order of genes within a pathway. Three situations are possible: sequential substrate product along an enzymatic pathway, successive structure formed during development of an organ, and third is the regulatory pathway that can have two outcomes. So, epistasis can be understood, or it goes by three pathways. Mutation in such pathways often have a distinct and opposite phenotypes, uninduced versus induced kind of fate. For example, if we'll talk about the sequential substrate product along an enzymatic pathway, so if a process is going in a way that precursor is converted to ornithine in presence of gene A, then this ornithine is converted to citrulline in presence of gene B, and then this citrulline is finally getting converted to arginine in the process with the help of gene C. Now, for example, if somehow the gene A gets mutated, or it gets deleted, then in that situation, obviously, whole of the process won't be able to form, because ornithine is required for gene B to convert it to citrulline, and then this citrulline will be converted to arginine. Same goes for enzyme B, and same goes for enzyme C, but all of the cases will give us different kind of an expression. Here we'll have the precursor only, here we can get the ornithine, here it will give us the citrulline. showing that three different kind of enzyme, three different genes are involved in this single process, and they are all in sequential manner, so that inhibiting any one of them can cause three different expression but not the final expression at it was required. So this was a Beadle and Totem's evidence for the one gene one enzyme hypothesis, because obviously three genes are converting these three genes are involved in converting from ornithine to citrulline to arginine. Successive structures formed during development of an organ. So genotype is wild type, phenotype is wild type, and it will give us the fate as it generally does. In case of the Lin26 genotype, vulva less phenotype will be there, no PNP cells will be there. In case of let23, vulva less expression will be there, 1 and 2 primary and secondary fates. Lean 26 and 23 valvular expression will be there, no PNP cells will be there, and Lean 26 acts upstream of let 23. So obviously it is the same in case of the expression is same as it happened in case of the Lin 26 type of genotype, in case of the Lin 26 and let 23, because when let 23 was alone it gave us the tertiary kind of a fate of the cells. Okay, but when they both were together, both the mutations were together, then Lin 26 got epistatic over let 23. In the above two examples, earlier block is epistatic. Next is the regulatory pathway. So this was an example of A and B both sequential substrate product along an enzymatic pathway, successive structure formed during development of organ. So it is that ways only. The process is almost same. Next is the regulatory pathway that can have two outcomes. Mutation in such pathways often have distinct and opposite phenotypes, uninduced versus induced fate. Okay, so regulatory pathway that can have two outcomes. Mutation in such pathways often have distinct and opposite phenotypes, uninduced versus induced fate. Okay. Wild type will give whatever it was supposed to give. In case of the let23 kind of a genotype, valvular situation will be there. All cells will follow the tertiary fate. So let23 is required for the primary and secondary fates. Let60, it is a dominant kind of a mutation. So multivalva situation will be formed, activates one and secondary fate, primary and secondary fate. Let23 and let60, in which let60 is dominant, so it will again form the multivalva situation as let60 acts downstream of the let23. Okay, so it is always a very interesting thing to see that although both the genes are present, but mutation due to epistasis due to one gene can affect the expression of the other genes also. So this is how the complete pathway was decoded. Lin3 activates Lin23, Lin23 let60, and then this let60 leads to Lin45 and then Lin31, and that is how the fates of the cells are decided. Okay, so this is a mosaic analysis. In mosaic analysis, they define the anatomical focus of gene action. Mosaic animals contains both wild type and mutant cells generated by spontaneous loss of free duplication and recognized by including a cell marker. Okay, so mosaic analysis is anatomical focus of gene action will be seen in case of mosaic analysis. Mosaic analysis can be generated by X-ray mutagenesis, injection of DNA clones containing wild type copies of YFG and the marker. So it is the mosaic analysis of Lin15. Lin15 mutations are recessive mutations that cause all PNP cells to adopt an induced fate, causing multivalva phenotype. Okay? Sorry. So the point of duplication, no loss, and it will be wild type phenotype. याद रखिएगा, this is lin 15 we are talking about. In case of the parent's progeny, if lin 15 is mutated, then it can lead to multivulva situation in the AB, the daughter of parent progeny. Again, the multivulva situation, ABPL or ABPR. If lin 15 gets mutated, they'll show often a wild type of situation. That means lin 15 does not play a role here. Then in case of P1, multivulva kind of a situation, P1 no AC, that means anchor cell. Multivulva situation, no loss and no anchor cells, the vulva less kind of a situation will be there. So this is just to show you ki how different expressions can be formed. Uh, this is called as the mosaic analysis. So unexpectedly, this mosaic analysis showed that lin 15 activity is required in cells other than the PNPs and the ACs. ठीक है. So as I was telling you about, these are the cells that are involved in the vulva formation, mainly this P6P. So signal emanating from hype 7 represses an interesting intrinsic vulva program 1 to primary and secondary fates in the pups, and AC signal counteracts this repression for 3 of the PNPs, allowing them to express induced fates. Okay, so this is all mosaic analysis. This is the lineage of vulva ki which cell will form what and how. This is again that major signaling pathway that is involved in the vulva formation. The EGF signaling is this is RTK RAS map kinase pathway, notch signaling, Wnt signaling pathway, and these are all very important pathways. You'll study about them in the coming weeks. Then the mapping with molecular markers. How the mapping can be done using molecular markers. How it can be done. So restriction fragment length polymorphism can be used. Variable number of tandem repeats can be used. Single nucleotide polymorphism can be used. So variable number of tandem repeats can be used because there are different number of repeats present inside the body of every organism. It is not same for two organisms, so it can help in mapping. Again, the restriction fragment length polymorphism gives us a restriction that if this enzyme is there, then only the cut will happen, otherwise it won't happen. So it restricts these fragments and then help us to understand the mapping pattern. And single nucleotide polymorphism is the change in the single nucleotide, a polymorphic situation on the single nucleotide. So these are the mapping methods for molecular biology. This is an example to show you about that. We're not going into these details. I hope you have understood it better from the lectures. So with this we come to an end to the uh, discussion of the lectures. I hope you understood it well. If anyone has any confusion, they can ask the question. Otherwise, we'll move towards the um, assignment questions and then the questions that I have brought for you guys. Any questions, anyone? So if you guys want to understand the C. elegans development, how these cells affect each other, if you want to understand it in detail, then you can do it. There are many videos available on YouTube for that, and very informative ones, so you'll be able to understand it better from there. Because this is one of the very important aspects if any one of you are preparing for CSIR net. This is an important aspect, so I would suggest go for those videos and understand it better. ठीक है. Any questions, anyone? Otherwise, we'll move towards the questions of assignment and then the one that I have brought for you guys. And I would request everyone to participate in the question answer round. It will help you only. You will understand it better. If you don't know, you can know what is happening and how it is happening. Okay. याद रहेगा आप लोगों को whenever you'll go for some kind of a competitive exams or something. ठीक है. Last 1 minute, otherwise we'll move towards the assignment questions. Any questions, anyone? Okay, let's move towards the questions then. Uh, ठीक है. So, first question is, which of the following methods can help you identify multiple additional genes functioning in a single pathway? Question number one. Write your answers in the chat box. No one will speak it loud so that everyone can participate. So write your answers in the chat box. Question number one. Which of the following methods can help you identify multiple additional genes functioning in a single pathway? Question number one, everyone. I hope question number one is visible to you all. Question number one, everyone. Which of the following methods can help you identify multiple additional genes functioning in a single pathway? Options हैं suppressor screen, blue white screen, chromosome walking and synthetic screen. Suppressor screen, blue white screen, chromosome walking and synthetic screen. Okay, so the answer to this question is 1 and 4. That is suppressor screen and synthetic screen. Both of these processes helps us to identify the multiple or additional genes that function in a single pathway. Okay, so it is suppressor screen also and the synthetic screen also. Next question, question number 2. That is, assume that you have identified a new gene called gene X. About 10% of the progeny from animals homozygous for a null allele of gene X do not survive because a lethal allele. You suspect gene X functions redundantly with another gene, gene Y. Which of the following methods is most suitable for identifying gene Y? बताइए इसका आंसर. क्वेश्चन नंबर 2. बाकी लोग जवाब देना नहीं चाहते हैं, इन्होंने लेक्चर्स लिए नहीं है। क्वेश्चन नंबर 2 एवरीवन, समझ में आया ना सबको क्वेश्चन? सबको समझ में आया क्वेश्चन? व्हाट आर दे सेइंग? नहीं भी आया तो आई एक्सप्लेन वंस। सो अज्यूम दैट यू हैव आइडेंटिफाइड अ न्यू जीन, ठीक है? फॉर एग्जाम्पल यू हैव सम जीन, आपने उस जीन की जब when you examined the progeny of that gene, so 10% of the progeny was found to be dead because of the null allele, because the the allele was lethal. Then what happened is, you suspect that gene X functions redundantly with another gene. So you thought ki there are more than one genes involved in this process or whatever function that gene was performing. So which of the following method is most suitable for identifying gene Y? तो यही तो क्वेश्चन हमने 1 में किया है कि एडिशनल जीन्स के लिए हम और क्या कर सकते हैं. सो आंसर इज अगेन सिंथेटिक स्क्रीन, बिकॉज सप्रेसर स्क्रीनिंग तो यहाँ है ही नहीं. तो द आंसर विल कम टू सिंथेटिक स्क्रीन. सो द आंसर टू क्वेश्चन नंबर 2 इज सिंथेटिक स्क्रीन. ठीक है? साइलेंट लिसनर्स, आई होप आप लोगों को भी समझ आया हो. ठीक. क्वेश्चन नंबर 3 देन विल कम टू, which of the following points would you keep in mind while designing and performing a suppressor screen? Which of the following points would you keep in mind while designing and performing a suppressor screen? Question number 3, everyone. Question number 3. Which of the following points would you keep in mind while designing and performing a suppressor screen? And a hint is there are more than one options which are correct for this answer. Anyone who wants to answer the question? Yes, so answer to this question is the mutant phenotype should be really visible. ठीक है? The mutant phenotype should be really visible. If you won't be able to see the mutant phenotype, how will you understand that the screen has happened? The function that it was supposed to perform was not performed. One thing. Another is the number of progeny at the F2 generation should be large. ठीक है? So the number of progeny at the F2 generation should be large, so that you can gather as much as different kind of mutations, or we can say different kind of alleles from that whole thing. Next option is the new mutation must follow Mendelian inheritance pattern, so that you can examine it better. ठीक है? Next question is, following are some of the steps followed, not in order, to identify a gene's chromosomal location and function. Choose the correct order. Now we have to choose the correct order. So the transgenic rescue with cosmids, correlation of genetic map and physical map near the gene of interest, identification of DNA lesions in the gene of interest in the mutant, identification of chromosomal interval, transgenic rescue with wild type DNA clones. So these are the five things. Now you have to put them in correct order. You want to put them in correct order. Yes, come on. I hope the question is visible to you guys. So put them in correct order. Following are some of the steps followed not in order to identify a gene's chromosomal location and function. So you have to choose the correct order of these steps that are involved in identifying a chromosomal location and function of a gene. ठीक है? So answer it. Put them in correct order. Anji, anyone who can put them in correct order? A, B, C, D. What would be the answer? Question number 4 I'm talking about, guys. No one wants to answer the question. Question number 4. Steps involved in chromosomal mapping. Basically, chromosomal location and function of a particular gene. So what are the steps? Shall you are saying question number 4th answer is option 3rd? That is what you want to say? Let's see. So the answer to this question is option number 4. That means first will be identification of chromosomal intervals, then the transgenic rescue with cosmids, then the correlation of genetic map and physical map near the gene of interest, then the transgenic rescue with wild type DNA clones, and last will be the identification of DNA lesions in the gene of interest in the mutant. Okay, so this will be your answer. Next question number 5. The two general mutant phenotypes observed with regards to vulvar development in C. elegans are first and second. Okay, so what would be the answer for 5th? What would be the answer in first blank? The two general mutant phenotypes observed with regards to vulval development in C. elegans. ये तो हमने पूरे लेक्चर में ही पढ़ा है. So I don't think इसमें किसी को भी चूकना चाहिए. बताइए question number 5th answer. The first option is question number 5th and second option is question number 6th. So first answer for first blank. What would be the answer? You can answer both of them together also, there's no issue on that. So question number 5th and 6th. What are the two mutant phenotypes that are observed in case of the C. elegans with regards to the vulval development? Yes, the answer is multivulva or vulvaless. वैल्यूलेस नहीं शालिनी, vulvaless. ठीक है, तो आंसर इज आइदर मल्टीवल्वा और vulvaless. ठीक है. नेक्स्ट क्वेश्चन. Next question is question number 7. Lineage analysis is suitable for identifying which of the following? Lineage analysis is suitable for identifying which of the following? बताइए लीनिएज एनालिसिस क्या हेल्प करेगी? Lineage analysis suitable for identifying which of the following? There can be more than one answers for this question. आप बताइए. Option 4 is correct, Shalini, but there can be more than one option. So what would be the other answer? Developmental parents of a cell, obviously that we can understand from lineage. Developmental potentials of a cell, that we can understand from lineage analysis. What other thing can be understood by lineage analysis? Okay, so other thing that could be understood by lineage analysis is the cell-cell interactions. Okay, it would be cell-cell interactions that can be understood by lineage analysis. Okay, now moving on to question number 8. Which of the following statements are false? First is AC induces, that is anchor cell induces vulval development. PNPs are pluripotent. Tertiary fate is the ground or uninduced fate. Hypermorph, neomorph, or antimorph are types of recessive mutation. Now tell me the answer. Very easy question. Tell me the answer for this question. Question number 8. Now they are asked for statements. That means there would be more than one answer. बताइए क्वेश्चन नंबर 8 का आंसर एवरीवन. Question number 8. Which of the following statements are false? Anchor cell induces vulval development. PNPs are pluripotent. Tertiary fate is the ground or uninduced fate, and hypermorph, neomorph, or antimorph are types of recessive mutation. There can be more than one answers for this question. So what would be the answer? बताइए भाई बताइए. Okay, so the answer to this question is option number B and option number D. That means PNPs are pluripotent, which is not the case. And last option, that is hypomorph, neomorph, or endomorphs are types of recessive mutation. Again, a false statement because it is a type of dominant mutation. Okay? So answer to this question is option number B and option number D. Next question, question number 9. Which of the following is true with respect to mutations that affect genes encoding enzymes of a metabolic pathway? Which of the following is true with respect to mutations that affect genes encoding enzymes of a metabolic pathway? So बताइए इसका आंसर क्या होगा भाई? Question number 8. Sorry, question number 9. 8 तो हमारा हो गया. Question number 9. Which of the following is true with respect to mutations that affect genes encoding enzymes of a metabolic pathway? Very easy question. I think everyone should answer this question. Very easy question and very good question. Question number 9. Are you sure, Shalini? I think you should check your answer. उन्होंने पूछा which of the following statement is true with respect to the mutations that affect uh, genes encoding enzymes of a metabolic pathway. So they've asked for a true statement, not a false statement. ठीक है? So answer to this question, question number 9 is yes, upstream mutations will be epistatic over downstream mutation, because if an upstream enzyme will not be able to function, will not be give a substrate to the second gene, the downstream gene, then the pathway will not be able to proceed. So obviously the upstream mutation will be epistatic over downstream mutation. Okay. Now, question number 10. Mosaic analysis is used to primarily study what? Mosaic analysis is used to primarily study what? Question number 10, everybody. Mosaic analysis is generally used to study what? Primarily used to study what? बताइए 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 भाई. Again, a very easy question. Mosaic analysis is what we are talking about. Is used to primarily study what? Mosaic analysis. We have four options: spatial patterning of the cells, spatial patterning of the cells, anatomical focus of gene function, anatomical focus of gene expression, gene interactions. ठीक है, तो बताइए इसमें से मोजाइक एनालिसिस किसके लिए यूज होता है प्राइमरीली? ठीक है, तो आंसर टू दिस क्वेश्चन इज anatomical focus of gene function. ठीक है, anatomical focus of gene function. Next question. Assume a new recessive mutation was obtained by mutagenizing a strain A. In this species, strain B is used for crossing with strain A for SNP based gene mapping. That means single nucleotide polymorphism based gene mapping, as several SNPs have been well established between these two strains. So, among the F2 progenies displaying the mutant phenotype will be. Again, I'm reading the questions. So, assume a new recessive mutation was obtained by mutagenizing a strain A. In this species, strain B is used for crossing with strain A for SNP based gene mapping, as several SNPs have been well established between these two strains. So, among the F2 progenies displaying the mutant phenotype will be. बताइए भाई. क्वेश्चन नंबर 11 आंसर बताइए. F2 प्रोजेनी का फिनोटाइप क्या होगा? Question number 11. I hope you understood the question. Tell me the answer. Question number 11. And again, I'm telling you, there are more than one answers. This question. Mm, mm, mm. No one wants to answer. That's okay. So I'll answer this question for you guys. So answer will be two options again, as I was telling you. A and C. Strain A SNP will predominate if the SNP is close to mutant alleles locus. One, and another is both SNPs will be equally represented if the SNP is far from the mutant alleles locus. ठीक है? So both will, if they'll be at equal distance, sorry, if they are far from the mutant alleles locus, so both will be equally represented, and if any one of them is near, so that will predominate. ठीक है? So with this we come to an end of assignment questions. These are the answers. So assignment questions are done. Now we'll start with the questions that I have brought for you guys. So question number 1. We'll take very general, very basic questions for you guys. Uh, till now, if anyone had any problems, we can wait for 1 minute. If anyone had any problem, they can ask. Otherwise, we'll move towards these questions. Okay, no one wants to ask anything. Let's move towards this question. Question number 1. A mutation that produces a phenotype in the heterozygous condition is generally classified as. A mutation that produces a phenotype in the heterozygous condition is generally classified as. Question number 1. A mutation that produces a phenotype in the heterozygous condition is generally classified as. Four options we have: recessive, dominant, silent, or suppressor. ठीक है, तो answer to this question is dominant. Dominant, if the mutation is dominant, then obviously it will be able to express itself in the heterozygous condition also. ठीक है. Next question: a recessive mutation is expressed phenotypically when. A recessive mutation is expressed phenotypically when. We have four options. One mutant allele is present. Both homologous alleles carry the mutation. The mutation is located in an intron, and the wild type allele is overexpressed. So again, the answer to this question is both homologous alleles carry the mutation, because generally recessive kind of mutation or expression is generally I'm telling you about, is expressed when there are in a homologous kind of a situation. ठीक है. Next question: a loss of function mutation generally causes. A loss of function mutation generally causes. A loss of function mutation generally causes. Options are increased activity of a gene product, reduced or absent activity of a gene product, formation of a new chromosome, expression of an unrelated gene. So all other options point towards gain of function mutation. Only one option says loss of function mutation, and that is option B, reduced or absent activity of a gene product. ठीक है. Next question: a gain of function mutation generally causes. A gain of function mutation generally causes. What are the options? Reduced gene activity, novel ectopic or increased gene activity, complete loss of gene, failure of chromosome segregation. ठीक है, तो all other options point towards the loss of function mutation. Only one option says gain of function mutation, and that is option B, which says novel ectopic or increased gene activity. So it is novel ectopic or increased gene activity is what happens in case of the gain of function mutation. ठीक है. Now let's move towards the next question. Next question. So what does this question? Question says a dominant negative mutation produces a phenotype because the mutant protein is. Question number 5, everyone. What are the options do we have? Enhances the wild type protein, interferes with the function of the normal protein, is never translated, is always degraded before translation. These are the options we have. So, what would be the answer? A dominant negative mutation produces a phenotype because the mutant protein is what interferes with the function of a normal protein. It interferes with the function of a normal protein. Okay. Next question. Question number 6. A haploinsufficient gene produces a mutant phenotype when? A haploinsufficient gene produces a mutant phenotype when? Haploinsufficient gene produces a mutant phenotype when? Question number 6, everyone. Options are both alleles are functional, one functional copy is insufficient for a normal phenotype, the gene is located on the Y chromosome, and the gene is expressed only in the mitochondria. So, yes, the answer to this question is one functional copy is insufficient for a normal phenotype. तभी तो वो haplo insufficient हुई ना. Next question. A hypermorphic mutation is a mutation that. What does a hypermorphic mutation call does? Hypermorphic mutation, everyone. Options are causes increased activity of a normal gene function, causes complete loss of a gene function, produces a completely unrelated function, prevents DNA replication. So the answer is causes increased activity of a normal gene function. ठीक है? Causes increased activity of a normal gene function. Question number 8. Again, a hypermorphic mutation is best described as. Although the question is same, but the language has changed. So, if you look at question number 8, then what would be the answer? Sorry, it's a hypomorphic. It's not same. It's hypomorphic. It's not hypermorphic. So, question is again different. Question. It is hypomorphic. A hypomorphic mutation is best described as. Question number 8, everyone. The options are a complete loss of function mutation, a partial loss of function mutation, a mutation with increased activity, and a mutation that changes chromosome number. What would be the answer? So, yes, the answer to this question would be a partial loss of function mutation. Okay, it's a partial loss of function mutation. Next question. A neomorphic mutation produces. a neomorphic mutation produces? Question number 9. Option to this question is a new or novel function, complete loss of function, reduced expression of the normal function, only a silent nucleotide substitution. So answer to this question is uh, I'm asking about neomorphic. Shalini, I think you should revisit the question and see the options. What would be the answer? A new or novel function, complete loss of function, reduced expression of the normal function, only a silent nucleotide substitution. Yes, the answer would be a new or novel function. ठीक है? A new or neo means new. So obviously it will be a new or novel function. So now I hope you understood the difference between hypermorphic, neomorphic, hypomorphic and antimorphic. ठीक है? Next question. In a complementation test, two recessive mutations fail to complement each other. This usually indicates that they complete the sentence. In a complementation test, two recessive mutations fail to complement each other. This usually indicates that they what? Question number 10. Question number 10, everyone. I'll read out the options. So, affect different genes, they affect the same genes, are both mitochondrial mutations, or are located on known homologous chromosomes. So, the answer to this question is affect the same gene. They affect the same gene, that is why they are not able to complement each other. Question number 11. The proportion of individuals with a particular genotype that expresses the expected phenotype is called. So, the proportion of individual with a particular genotype that expresses the expected phenotype is called. Is called what? The options are expressivity, penetrance, pleiotropy, and epistasis. Pleiotropy and epistasis, you guys know. So the answer to this question is penetrance. Okay? So answer to this question is penetrance. Next question. The degree or severity with which a genotype is expressed is called. Now they have asked a degree or severity with which a genotype is expressed is called. Question number 12 we are talking about. Question number 12, everyone. The options are penetrance, expressivity, dominance, and complementation. So, you know what penetrance is, you know what dominance is, and you also know what complementation is. So, the answer to this question is expressivity. It is the degree with which a genotype is expressed. Okay? Now, question number 13. Epistasis occurs when? Epistasis occurs when? Question number 13, everyone. Options are: one gene modifies or masks the effect of another gene. Two alleles occupy different chromosomes. A gene is expressed only in males. A mutation affects only RNA processing. So, last two options have nothing to do with that. So, obviously, the answer is A. One gene modifies or masks the effect of another gene. So, this is what epistasis is. Question number 14. In a double mutant analysis, the phenotype of the double mutant resembles the phenotype of mutation A. So, mutation A is usually considered. Question number 14. In a doubled mutant analysis, the phenotype of double mutant resembles the phenotype of mutation A. So, mutation A is usually considered. Question number 14. Question number 14, everyone. I'll read the options. It is downstream of mutation B. It is epistatic to mutation B. It is complementary to mutation B and unlinked to mutation B. These are the options. So the answer to this question would be a double mutant analysis. The phenotype of double mutant resembles the phenotype of mutation A. So mutation A is usually considered. Answer is epistatic to mutation B. ठीक है? Epistatic to mutation B. Question number 15. If gene A acts upstream of gene B in a linear pathway, the phenotype of an A mutant, B mutant, double mutant usually resembles. Question number 15, everyone. I'll read out the options: the wild type phenotype, the A mutant phenotype, the B mutant phenotype, and a completely new phenotype. What would be the answer? Question number 15. Okay, I'll answer the question. Answer would be the A mutant phenotype, because A is upstream of B. So if A is mutated, B will obviously not be able to function. So the phenotype would be exactly similar to what A mutant would have looked like. ठीक है, तो answer to question number 15 is A mutant phenotype. Question number 16. In a recessive epistasis interaction, the homozygous recessive genotype at one locus does what? In a recessive epistasis interaction, the homozygous recessive genotype at one locus does. The options are: enhances the expression of a second locus, masks the expression of a second locus, always causes lethality, or produces a dominant phenotype. So the answer to this question is masks the expression of a second locus. Okay, it would mask the expression of a second locus. Question number 17. The typical F2 ratio of a recessive epistasis is, although we haven't discussed it in the lecture, but if you guys have studied it, then it would be great. Otherwise, I'll tell you the answer. Anyone would like to try, or I should tell you the answer? Yes, answer is 9 is to 3 is to 4. Well done, Shalini. Next question is, the typical F2 ratio for dominant epistasis is. Again, something we haven't discussed till now, but if anyone wants to try. Question number 18. The typical F2 ratio for a dominant epistasis is. Yes, it is 12 is to 3 is to 1. Next question. Complementary gene action, also called as duplicate recessive epistasis, commonly produces the ratio. Yes, it is 9 is to 7. Next is duplicate dominant epistasis commonly produces the ratio. Question number 20. Duplicate dominant epistasis commonly produces the ratio. Yes, it is 15 is to 1. So with this, we come to an end of the questions that I have brought for you guys. So if anyone wants to ask any question, any query, any confusion, then they can ask. Otherwise, we can stop the meeting here. Thank you all for joining. You guys can leave the meeting if you don't have anything to ask. We'll meet next week on Tuesday again at 6 p.m. Question number 10. Let me see. Question number 10. Okay, so in a complementation test, two recessive mutations fail to complement each other. This usually indicates that they affect what? They affect the same gene. So what happens is, complementation means what? Ki if uh, one allele or one gene is not able to perform the function, then the other allele or other gene will help to perform that wild type of function, or whatever the function that gene was supposed to perform. But if that complementation fails, that means that the both the alleles, या फिर if we'll say both the mutation are occurring on the on both of the alleles that were there to complement each other. So obviously that means that both of the alleles were a part of the single gene. That is why they were not able to complement each other. Did you understand what I told you? Let's take an example. For an example, there's gene A, which is controlling the expression of any trait, any kind of a trait in an organism, and mutations happen. Now we have an allele capital A and a allele small a for that expression. Okay. Now they are saying कि two recessive mutation fail to complement each other. That means one gene gets mutated with the recessive function, then obviously the other dominant one will perform the function and it will be complemented, the expression will be there. But if both of the alleles get mutated, both of the alleles undergo the same mutation, the recessive mutation, then no complementation gene will be present, nothing will be there to complement this mutation. So obviously this will affect, this shows that the effect is on the same gene, that is why nothing was there to complement that expression. Now did you understand? Okay. If still there is some problem, read about complementation so you'll understand it better. Still some confusion is left, we can discuss in the next class also. There's no issue in that. Okay, guys. Thank you so much for joining. You guys can leave the meeting now. Bye. Good night.